which would be fantastic. So,
Yeah.
but I'm going to, obviously, this is my day job now. I'm on my own research on what we're doing in the lab. And I'm very happy to give you the story of inflammasome, because Maeve mentioned that. And I'm also going to talk more broadly about inflammation and inflammatory diseases and where I see the whole area going, I guess. So the title is Adventures. I've spent 35 years since my PhD working on the inflammatory process. I began working on cytokines way back in the mid-80s, before taxi drivers knew what a cytokine was. I worked on IL-1, TNF, IL-6, all the big ones. This is the late 80s. We knew about those cytokines back then. And my research has always really been about trying to understand the molecular basis for inflammation. Can we figure out what the molecular pathways are? And then can we then understand them in a disease context, and then maybe develop ways to block them? And that's been the passion of mine for the past 35 years, I guess. And I am going to talk about COVID a bit, because COVID is an inflammatory condition, and we've got a couple of results on COVID as well I want to share with you. Now, I might start with this though. So this is a big translational day, of course, and these diseases are massively important. I mean, there is still a huge unmet need to treat inflammatory diseases with great efficacy. Now, this slide here captures the challenge in a way. Rheumatoid arthritis, RA, IBD, that's like Crohn's and colitis, MS, just to name 3. But I could list about 60 diseases that are classified as inflammatory. And there are therapies out there. Anti-TNFs work well in rheumatoid, for example, and also to some extent in IBD. In MS, there's various therapies as well. But the truth is they don't work great. They do give some benefits to patients, of course they do. But in the big pharma world, they talk about this efficacy ceiling. And can you get drugs to breach that? And, you know, drugs, for example, that target cytokines or T cells, abatacept is a T cell drug, rituximab targets B cells. You get some respond very well, but on average, you get about a 50% response. Some patients stop responding. So there's a big need to break that ceiling. And my lab moved increasingly away from what you might think of as traditional immune inflammatory pathways. And we work on 2 big things, NLRP3, which I'll mention to do with inflammasome, and metabolic change. In the past 10 years or so, my lab has kind of shifted more towards those things. And metabolism wouldn't be seen normally as part of any immunologist's interests. But what's happened in the past 10 years is remarkable. All these great new insights into metabolic pathways, changing, say, in a macrophage or a T cell, and that presents brand new opportunities. And in many ways, what this slide captures is, if you want to break that ceiling, you've got to go beyond the traditional immune targets and go for something else that might actually work better, or indeed work in combination. And we're very interested in combination therapies in all kinds of ways. So the 2 things I'm going to talk about mainly are NLRP3 and targeting metabolism. Now, NLRP3, first of all, it's a striking business how this sort of emerged. It's a central sensor of what you might call damage. Now, damage can mean infection, microbial attack. It can mean sterile damage. There's things called DAMPs, which come from damaged tissue, or PAMPs from microbes. NLRP3 seems to sense these. And in all these different tissues, you see NLRP3 being overactive in certain disease situations, mainly in macrophages. And the reason is it senses what you might call noxious things in the macrophage. And this can be infections, of course, but it's also things like cholesterol crystals or an amyloid factor called IAPP in type 2 diabetes. That was my first paper on this back in 2010, actually, was on type 2 diabetes. And then beta amyloid, you all have heard of that, I imagine, in Alzheimer's, alpha-synuclein in Parkinson's, uric acid in gout. And what's happening is the macrophage, its day job is to engulf these things and clear them. That's what macrophages do, and cause a bit of inflammation to cause, like, more blood flow, bring in more macrophages, bring in certain factors to help the process along. And then these things are cleared, and then the situation is resolved. The trouble is these go out of control in so many different diseases, and we still don't fully know why. But cholesterol, it could be high cholesterol diet. Why does beta amyloid get deposited? We don't know. So it's a bit of a mystery, but we do know that NLRP3 seems to sense all these different things. And what that means is that literature joins many other pathways in a way that explain inflammation. And we call it autoinflammation, by the way. Say, for example, in gout, where there's no T cells, but there are macrophages, it's called autoinflammatory. Equally, the autoimmune situations, you will see macrophages, but you'll also see T and B cells. So there's a bit of a division between those 2 disease types. But these 4 pathways, in many ways, explain the pathology in many of these diseases, and many drug companies are going after them. In the case of autoimmunity, it's the JAK-STAT pathway. In the case of autoimmunity as well, it's CGAS, sensing DNA, type 1 interferons. In the case of NLRP3, it's noxious different stimuli, and that will drive IL-1, a very important cytokine, comes off this pathway, IL-18, and these things called gasdermins, which cause a type of cell death called pyroptosis. Back to that a bit later. And then the TNF pathways, RIP kinases are a major player there, again driving cell death. So what's striking is it's coming down, and again, I've been in this business for over 30 years. It's coming down to 4 main pathways, actually, that may be the basis for so many different diseases. And of course, inhibitors are now being developed. Now, there are JAK inhibitors already approved. There's a drug called Xeljanz for rheumatoid arthritis, for example, another one called baricitinib. These block JAKs, and they have anti-inflammatory effects, and they're being used in the context of inflammatory diseases and in COVID as well, actually, which I'll come back to. So they're on the market already, many more being developed though. CGAS inhibitors, they're in development, RIPK inhibitors as well, and then the NLRP3 inhibitor program, and that's where my lab had a major focus beginning in 2010. And to give you a little snapshot of the sector as a whole, because this is a translational meeting, Jeremy Skillington, who was a key guy in inflazome actually, sent me this analysis. It's by a crowd called Torreya. They're one of these big biotech analyst firms, big biotech uptake 2021. And this, now look at this to get your heads around. These are all the big targets in inflammatory diseases and the different companies and the different stages. It could be approved, it could be clinical. And look, the IL-1 inflammasome pathway is really important here. Here's the JAK-STAT pathway. Here are many different drugs being developed. So it's a massive area for the industry. And all these different molecules are now being tested in these different diseases. Now, how did we get to it? And how did we come to be a player in the NLRP3 world? Well, it's my lifelong love affair with IL-1 because I began my PhD in 1985 working on IL-1. And that's a key inflammatory driver. And then we got inflazome going to leverage, I suppose, some of our work on this pathway. And I'm going to give you a little bit of history of this because people are interested in seeing how we got from my lab into inflazome and then beyond. And of course, inflazome was then acquired by Roche. As Maeve said, I'll come back to that towards the end. But look at this, it goes back to 1986. Now, I was asked to give this talk in Roche back in November to announce the deal with the 2 companies, and I was asked to give the overview of the history of inflazome. And 86, I'm at a conference in London. I did my PhD in London. And a guy called Charles D. Neill gets up and he gives a talk on IL-1. And it's one of those moments when I remember vividly where I was sitting because he gave this fantastic talk. And he said, IL-1 is in all these different diseases. They could be metabolic, they could be in the CNS, they could be hematologic, they could be in the vasculature, and there'll be many different diseases, hemodynamic effects as well. And I remember vividly thinking, this is a good molecule, I got to work on this because it seems to be central to so many different situations. And then the field began to wonder, how does IL-1 get made? It's made as a pro form, first of all, in response to things like TLRs, which my lab had worked on. NF-kappa B drives this. And then an enzyme called caspase 1 cleaves it to make the mature form, and now that causes all the inflammation. And it was very clear in the sort of late 80s, early 90s, a second signal was needed to drive caspase 1 to process pro-IL-1. And many things were shown to do this. Now, this is my list of noxious things. It could be uric acid, it could be whatever. The big question is, what was that bit missing there? And these 3 people, Jörg Chopp, Hal Hoffman, those 2 in particular, and Alan Wanderer, they get the credit for discovering NLRP3. And Hal Hoffman finds a gene mutated in a very rare autoinflammatory syndrome called Muckle-Wells, and he names the gene cryopyrin. That means hot and cold. These people get fevers and they get chills. That's the same thing as NLRP3 because Jörg Chopp comes along and he names NLRP3. And in 2001, I'm at a conference in Hawaii. It was a tough old life in those days, traveling around the world. It isn't possible anymore. And he gives a talk on a thing called the pyrin domain. And I gave my talk next, and his talk was mind-blowing. That's my second moment in the audience. And he says a thing called the inflammasome. It drives processing of IL-1. So he'd found, and Hal between them had found NLRP3 as the key driver of caspase 1, sensing all these different things. And in essence, this is what it does. And this is my own lab. Annie Curtis used to be in my lab. She took this EM of macrophages a few years ago now. They all have NLRP3 in them, and they sense damaged tissue, insoluble aggregates of proteins, metabolic products. And what this is actually about then is frustration. And there's a good slide to show that. So the macrophage tries to take up these things as its job as a phagocytic cell. NLRP3 senses them and it gets very frustrated, and it might die, and it might make IL-1. And the key output, of course, is IL-1. Downstream of these are caspase 1, and you get IL-1, and IL-18 is processed, and these gasdermins, they get processed as well, and they drive cell death. So suddenly a whole world opens up in the past, 6, 7, 8 years now to fit in those details. And again, back to my slide, we're sensing all these noxious things through NLRP3. And then the hypothesis that we would have derived probably in 2012 now everybody said this, it wasn't unique to us. It's a domino effect, noxious things, NLRP3, IL-1, IL-18, cell death, pyroptosis type of cell death, inflammation, and could this involve eventually driving all these different diseases? And then in 2012, I think it was, we find an inhibitor of NLRP3. So my lab was looking for inhibitors. My previous company, Opsona, tried to find them, spent €2 million, couldn't find one. That was my company. My academic lab stumbles into this compound here. And it was called CRID3 originally. It had been found by Pfizer back in 1997, again, serendipity in a way, as a molecule to block IL-1 production. And they stopped working on it for various reasons. And I wondered, well, it blocks IL-1. Could it be blocking NLRP3? Lucky question to ask. It may not have been an NLRP3 inhibitor. And lo and behold, this turned out to be what everybody's looking for, a very specific NLRP3 inhibitor. And here's some data for you. There's uric acid driving IL-1 production. NLRP3 inhibitor blocks. So we have to rename it MCC 950 for various reasons, but it is the CRID3, the Pfizer compound. Blocks IL-1, didn't block TNF, that's a control. TNF doesn't come off this inflammasome, so that was quite nice. We had our paper 2014 or 2015, our paper comes out describing a small molecule inhibitor of NLRP3 for multiple diseases. And then, of course, we knew, let's make this even more interesting. And we gave the compound out then to over 50 labs. You couldn't buy it in the world, it wasn't available. Matt Cooper, my key chemistry collaborator, who is my co-founder of Inflazome, he gives it out to all these labs, and the results just began to come in. And this compound, this single, I'll show it to you again, the structure. Can you imagine this little arrangement of carbons and nitrogens worked in 80 models of inflammation: Parkinson's, Alzheimer's, ALS, gout, acute kidney injury, dermatitis, MS, atherosclerosis, NASH, stroke, MS, colitis, psoriasis, and even in some infectious diseases where there's overactivation of inflammation. It's striking. 80 models this compound worked in. Now, again, that was predictable because NLRP3 have been implicated in all those diseases, either through overexpression or knockout mice being protected. So we were asked for the compound by labs expert in those areas. But it's a striking business that this single molecule can work in all these inflammatory diseases. And our paper comes out, and very nicely, Jörg Eder, head of immunology inflammation at Novartis, he said, oh, Neill's work is a game changer. I'm not smoke up my own ass there, but he said, look, this is now tractable with a small molecule, and we got there. Isn't that great? No drug company, they're all looking for it, they couldn't find it. Dumb luck, we found this very specific NLRP3 inhibitor. Now, what do we do next? Well, Pfizer had dropped it because they got a bit of tox in a rheumatoid arthritis trial, was the rumor. So that CRID3 compound wasn't going to make it to the clinic. And Matt Cooper said to me, to give him credit, he said, let's go for it. Let's try and make derivatives. And I said, but surely to God, Pfizer have done that. They own the original molecule. He's no, he says, we'll beat Pfizer. And this classic Australian, he's from Australia, you know, there's confidence. And I said, oh, okay, let's go for it. So we decided then to form our company. We raised money. Jeremy comes in as our key business development guy. We raised 45 million in the end. That was me selling the idea to all these investors. And everybody knew this could be great, but it's very risky and things go down all the time. We raise money, we make derivatives, we build a team. Matt's big strength was he leaves his academic post actually on sabbatical or a leave of absence, comes to Ireland and then builds the team. 14 people we recruit, all kinds of things here, business development. We build the company, and I'm the chief scientist and Matt is the CEO. And then, long story short, we find two molecules. All the chemistry works. We call them IZD174, IZD334, based on CRID3. We own the patents. Very potent, very selective, passing all the phase 1, did a phase 1 trial, did lots of tox testing, did one little tiny phase 2 trial with this rare disease. And then, of course, what happened next? We go and sell the company. Now, we could have kept the company going. That would have meant raising probably 300 million to go to a phase 2 trial. say in colitis. You can't raise that kind of money. No venture capital to give you that. Your only option is to get bought by a big pharma. We've got very deep pockets. We're on the road now for 2 years trying to flog this to companies. We made hundreds of presentations over those 2 years to companies. And finally, Roche bought us after a lot of 9 months of diligence. It was incredible. And they buy us, and that was last September. Now, they buy the 2 molecules. And look at this, the banker was Lazard, anybody who's commercial, and they made money on the deal, and they made us all these little crystal things as a memento. That little ball thing, that's NLRP3, the structure of it, you know? So it's quite nice to have that as an artifact. Now, what does this mean for us? Well, the great news now is, if I go back to one of these slides here, Inflazome can now realize its potential. Roche clinical trials in 6 huge diseases with our molecules. That's why it's good. Now, obviously, money came in and we all made a bit of money. The investors made a huge amount because we got a big return. But the fact is, it's joyous because Roche will now spend $1 billion on this money and take it to the clinic in these huge indications. And now I'm continuing on as a consultant, which is nice. I advise them. And I'm waiting with bated breath now because no more than COVID, we're waiting for trials. So the trials will start very soon. So it's very exciting now that we're going to really test this in humans, that this is going to work. Now, the second part, what time is it? Is how long have I got, Maeve? We'll probably have another 10 minutes, haven't we?
We'll go until 1:15 if that's all right.
Oh, perfect. Yeah, great. Okay. Now, the second half is the newer stuff. So what I'm doing now is, by the way, to give you my life in this regard, it's with Roche. It's realized what we can do with it. Now, I'm talking about Parkinson's, Alzheimer's, colitis, Crohn's, you know, maybe atherosclerosis. They're running those trials. It's great. And we wait and see what happens. Now, meanwhile, by the way, to fill you in, after our paper came out, 3 other companies were founded making molecules based on that Pfizer compound, getting around the patent. There's about maybe 20 NLRP3 inhibitors now in development. Novartis bought one, a company called IFM. So we're going to see several NLRP3 inhibitors in development, which is exciting. Let's hope ours makes it, I suppose. Maybe, you know, you never know. But certainly now it opened up the field in that sense. Someone else might get there ahead of us. Now, second thing we're doing, though, that's really relevant to the topic today is other targets, I guess, other approaches. Now, the one thing that three of these 4 pathways have in common is mitochondrial dysfunction. So mitochondrial DNA in a damaged cell, C-gas will sense it. NLRP3 is actually sensed by ROS, reactive oxygen, coming from mitochondria and DNA. Again, mitochondrial dysfunction drives the RIPK pathway. So this got me.
Sorry, Luke, I think you might be muted there. I'm not sure what's after happening. I've just asked to unmute you there.
There we go. I'm back.
Yeah.
Great. Okay. Did you mute me on purpose there, mate?
I didn't mute you at all.
So the bottom line from this one is I knew the mitochondria were disturbing in this process, and I got back into metabolism. I'm a biochemist originally, so it was easy enough for me to re-enter metabolism, I guess. Now remember, the mitochondria, there's a long literature on them involved in immunity. The first was cytochrome C release, a fantastic discovery when you think of it. Unexpected electron transport chain compound driving apoptosis, you know. And then the MABS pathway, which involves RIG-I. Anybody who's an aficionado will know what that is in response to viral RNA, very relevant for COVID. That will drive NF-kappa B in the IRFs. Mitochondrial DNA, TOL9 can sense that, and C-gas can sense that, and NLRP3 with ROS. So mitochondria now are the powerhouse of immunity. That was a review I did recently with Ivana Mills. So mitochondria suddenly are more and more interesting from an immunological point of view, as opposed to just, say, a bioenergetic point of view. And these 2 pathways then, there's apoptosis, that's one type of cell death. Pyroptosis is the other, driven by NLRP3. Here's a good way to look at that. Isn't that a good stuff? So pyroptosis, the cell explodes and releases all these noxious things. Apoptosis, much more controlled, as I hope you all know. So it's a very important pathway, this pyroptotic pathway. Now, to give you a little bit of what we're doing on this at the moment, we begin working on metabolism. We make lots of discoveries, lots of potential for glycolysis and all kinds of things. 2 things that we're really working on is Krebs cycle, because it turns out that becomes interesting in a macrophage during inflammation. And I'm going to give you 2 quick stories, one on succinate, the other on itaconate. There's Krebs cycle. You'll all remember it, I hope. You would have learned it as electron transport chain. But remember, amino acids, purines, lipids, it's the central metabolic hub. This is how you might remember it. Learn the Krebs cycle, forget the Krebs cycle, and that becomes a cycle that goes on forever. Now, the two metabolites, I want to talk about succinate, which is in Krebs, and itaconate. Now, this is very important when it comes off Krebs. It's a diversion away from Krebs, and we have discovered, and several labs, these two metabolites have a critical role in immunity and inflammation. Now, we published a lot on succinate. It comes out of mitochondria, it drives HIF, and HIF will drive all these HIF-dependent genes that are in inflammation. Reverse electron transport happens, and that drives ROS. There's also a receptor that drives chemotaxis, for example. Here's reverse electron transport. We did a 2016 paper showing that the electron transport chain runs backwards in an inflammatory macrophage. Can you believe it? And that drives reactive oxygen, and that's inflammatory. That was one thing we published. And then itaconate, 2018, our paper came out, explosion of papers on this, with this metabolite involved in the immune system, but also to do with pathogens. And there's a massively growing literature, and also viruses. And towards the end, you'll see why this is relevant. Itaconate can regulate type 1 interferons. It can block Zika virus. It can block influenza. It can affect VSV. These are recent papers in the last 2 years. So there's something very interesting about this metabolite. It can target viruses. It can also target bacteria. This paper here is on Pseudomonas. It turns out Pseudomonas eats this stuff, and that's relevant for cystic fibrosis, for instance. There's a role for this in host pathogen interactions. And in 2016, we saw massive increase in itaconate in macrophages with LPS. LPS is a gram-negative bacterial factor we use a lot to drive cells. It builds up massively in macrophages, and that was a novel observation. 1 or 2 labs had seen this as well, and we jumped on this very small group of labs that began working on this. And then we get more and more interesting data. What does it do? It's anti-inflammatory. If we give itaconate to mice and then give them LPS to create sepsis, this is like gram-negative sepsis, we can protect the mice. This is a metabolite coming off Krebs cycle, remember, tiny metabolite is protective in LPS-induced sepsis. The enzyme that makes it IRG1, that takes a connotate from Krebs to make itaconate. If you knock down that enzyme, itaconate falls and you boost IL-1, back to my favorite molecule. Now again, that's because itaconate is limiting IL-1. So you lose the enzyme, you boost IL-1. A couple of recent papers that are fantastic. They've measured itaconate in rheumatoid arthritis. There's an inverse correlation with disease score. Now what that means is the lower the itaconate is, the higher the disease score. Here's itaconate along here. Low levels of itaconate, higher disease score. As itaconate goes up, the disease goes down. That's a correlation with patients. You see an inverse with CRP, actually, which anybody's interested in rheumatoid. And then this paper got me really excited. Came out in August. A big metabolomic screen of COVID-19. And guess what? Yet again, an inverse relationship with severity. So in more severe patients with COVID-19, itaconate levels are low. And then we're thinking that's because, you know, there's not so much an anti-inflammatory effect. You're losing a key anti-inflammatory metabolite and the disease gets worse. That got my imagination because obviously we're all obsessed with COVID now. Now, it's anti-inflammatory. What does it do? New thing, it modifies cysteines on target proteins. We reported 30 proteins in our paper in 2018, directly modified by itaconate. Glycolysis gets modified, I think allanexin A1. And then that was confirmed, thankfully, by other labs, which is always a relief when your work is reproduced. A Chinese group, they find lots of proteins modified. Glycolysis stands out. Aldolase, GAPDH, and LDH are all modified by itaconate. Now, glycolysis, by the way, is pro-inflammatory. It's part of the Warburg effect in macrophages. So that was good because it would suggest an anti-inflammatory mechanism. And now I'm going to give you 3 little stories on our most recent work. 1 published, 2 not published yet. My lab, I think about 10 of them now are working on itaconate. So we found some very interesting new things. First of all, guess what? It blocks my favorite pathway, NLRP3. And here's ATP driving IL-1, blocked by itaconate. There's nigericin. These are ways to drive NLRP3. Here's IL-18, profoundly blocked by itaconate. Here's IRG1 knockout mice. They can't make itaconate. They make more IL-1 because you're losing that break, and that's very nice because that shows endogenously. And then we got some samples from these rare patients with Muckle-Wells syndrome. Remember I mentioned it earlier? They have a mutation in NLRP3. It's overactive in those patients. Ex vivo, we can block all the IL-1 with itaconate. Look at that. That's a really nice translational data there. That's patient samples for blocking IL-1. What's the target? I mentioned cysteine modification. And lo and behold, it blocks NLRP3 by targeting Cis548. We did this by mass spec. So another substrate for itaconate with cysteine modification. And we were looking at the cover of Cell Metabolism about 3 months ago. So itaconate targets NLRP3. Now that just tells you potentially how important itaconate is. It's an endogenous way to block NLRP3. It's a bit like our Roche collaboration. They're chemicals, they're pharmaceuticals. Here's a natural metabolite that can block a very important pathway. Now, secondly, JAK inhibitors. Now, earlier, did I mention JAK stats? I did. Now, it turns out the macrophages can release itaconate. There was evidence it goes into a cell type called M2 macrophages, and that got us going on this. And lo and behold, it can block JAK1, that kinase. Now, JAK1 is used by IL-4 and IL-13, and here we see IL-4 driving various target genes, and there's itaconate blocking them. So IL-4 is driving Rh1, say, dose response inhibition there. It also blocks IL-13, another very important type 2 cytokine. Now, if you're an immunologist, you know what I'm talking about. IL-4, IL-13 are very important for allergy and asthma. They're very inflammatory in the airways in that context. They go through the JAK-STAT pathway. And here we see, again, itaconate very nicely blocking IL-13 there with 3 different readouts. What's the target? We looked at JAKs and STATs, and look at this. It's blocking STAT phosphorylation. There's itaconate compared to control, blocking phosphorylation of STAT6. Here's JAK1, which is on the JAK-STAT pathway. Again, itaconate in the Western. This is phospho-JAK blocked by JAK1. And then finally, we went in vivo. Oh, no, sorry, by the way, we also found it modifies a cysteine on JAK1. That's the important thing. Our Chinese collaborator, Chu Wang, showed that. So it's cysteine modification. Here's the big modification. There's JAK1 being modified on a cysteine by itaconate compared to control. So it is a cysteine modification. We went in vivo as well. We like to go in vivo just to make sure. It's tricky with metabolism. You got to go in vivo as well. Cells in culture are kind of metabolically stressed. You can challenge mice with a thing called IL-4C. That drives JAK1 STAT pathway in mice. And again, gene expression, itaconate given to mice will block all those IL-4 driven genes, which is very nice. And phospho-STAT6 is blocked as well, and phospho-JAK1 in vivo. So itaconate, we think it's targeting JAK1. And that could be very interesting because JAK1 is a key inflammatory pathway in different contexts, interferon beta, Th2 cells as well for the aficionados. And it does it by cysteine modification. And there is a JAK inhibitor already out there called ritlecitinib that blocks a cysteine on JAK3. So this is not unusual to see cysteine modification. Now, finally, COVID. How do I do this? Well, again, no more than many of us in this business, we can switch into COVID very easily. Now obviously enough, I wasn't working on COVID much because it hadn't been discovered, but also my main interest was never infectious diseases. I would have done stuff on chikungunya virus in the past, actually, something a bit on vaccinia, but in general terms, my lab worked more on inflammatory diseases. But it became very clear early on, NLRP3 is in COVID and in the macrophage, in the lungs, you know. And then there was the lovely Jex Med paper, big activation of NLRP3 in COVID patient samples from peripheral blood, for example, and in lung tissue, this pink spot, that's all active NLRP3. So of course, my lab then began to wonder, we could do work on this. And we measured IL-18 ourselves in our samples from James's with our clinical collaborators. Big increase in IL-18 with severity compared to non-severe patients. And remember, this is coming off NLRP3, so that evidence gave us even more reason to go after this. And then I told you already, a decrease in itaconate with severity was reported. So we began wondering, would this be of interest? And also, this paper came out on succinate and SARS-CoV-2, reverse electron transport, just like we'd shown with LPS and macrophages. You get HIF, you get glycols with COVID-2. So again, it was easy for us to switch into this. And then there was this literature on itaconate killing Zika. So this could be COVID. And remember, it's anti-inflammatory. And we know dexamethasone is showing benefits in COVID. That's another anti-inflammatory agent. So we start testing it hot off the press. Here is itaconate protecting cells from dying from SARS-CoV-2 infection. These are lung epithelial cells. You infect them, they die. You give them itaconate, you stop them dying. And that seems to be an antiviral effect. We measured RDRP that the polymerase, again, expression was suppressed by itaconate in SARS-CoV-2 infected cells. Now we got scooped immediately, a paper in Nature Communications, itaconate targeting SARS-CoV-2. So we're in the right direction. Christian Holm, we know his lab very well. So it looks as if itaconate can limit this virus. Finally, our Leuven collaborators, there's a hamster model of COVID-19 and looks just like human disease actually. And you get a big lung pathology, all these different scores. And here's the infected mice, big increase in scores for inflammation in the lungs. We gave the hamsters itaconate, it blocked lung inflammation. Now this could be blocking NLRP3. It could be antiviral. We don't know the exact, there's number of targets here, but we're now, you can imagine now we're going hell for leather after this. What are we doing translationally? We've got derivatives of this molecule as well. My company Citrix, which are much better than the endogenous molecule. Secondly, dimethyl fumarate is a compound that's in the clinic already for MS that mimics itaconate. Now, finally, let me give you the overarching summary. I think I've given you one big idea here. I hope targeting cysteines might be anti-inflammatory, and things like itaconate and fumarate will target cysteines and inflammatory proteins and inactivate them. And there's reports on things like GAPDH and glycolysis, gasdermins. Drugs do this as well. Dimethyl fumarate can target cysteines. So can disulfiram, serendipitously. That's used for alcoholics, actually, but it targets cysteines. And then we got 401. So here we have a new way to targeting NLRP3. JAK1 could be in this category as well. So it's a new idea to block inflammation by targeting cysteines on important inflammatory proteins. And there are drugs in the clinic already. Bruton's tyrosine kinase inhibitor does it, ritlecitinib does it. So these already target cysteines. So the overarching idea here would be, here's a new way to target inflammation that could be applicable to many different inflammatory diseases, including COVID-19, selective targeting of cysteines. And we're kind of piggybacking on a natural process with these therapeutic agents is the idea, I suppose. And who knows, this could be another way to block inflammation, just like the NLRP3 program I told you about, to decrease inflammation and resolve the inflammatory process. So my lab at the moment is now hell for leather, you can imagine, going after these aspects of cysteine modification. And it'll give us something else to fire drugs at, another process to target, I suppose. And I'll finish there and thank the people in the lab, obviously enough. Alex, great job on NLRP3, very grateful to him. Mara did all the work on JAK1, actually. Hauke is helping with the COVID work. And of course, very important collaborators, Mike Murphy in Cambridge is a key advisor for much of this work. Richard Hartley is the chemist who helps us with 4OI and derivatives. Roman Fisher helps with cysteine modification. Martin van Hemert did the SARS infection model that we have in Leiden, and Laurens, Leisenberg, and Leuven. They're helping with the in vivo work with COVID-2. We have got COVID-2 in our lab in TVSI and Trinity. We're just beginning to work on the real virus. So at the moment, we're just collaborating with those labs to get some work done on that area. And then finally, I'd like to thank our funders, SFI Welcome, and the European Research Council. Thank you very much.
Thank you very much, Luke. That was really insightful and informative into your journey, which is great to hear. Such a success story as well, and really interesting then to hear your most recent developments as well. I just have a question about, so say you're talking about the recent and really exciting results you have, say for example with aticarate, sorry, I'm probably
we pronounce it 3 different ways ourselves, to be honest.
I'll be pronouncing it a different way in a minute now, but and how would you go, or how would, so say for students here listening who have experience working in the lab, you know, maybe we're not making these major discoveries, but how are you going to approach now, I suppose, using those discoveries that you found and trying to create them as therapeutic targets and taking them to the next stage? That's
the key question. More in general for your whole talk, our whole talks today are similar theme. How do you validate a target? How do you make a discovery and translate that into a new medicine? That's the key thing that we're in our minds all the time. There's a lot of work going on around the world in itaconate, and it's all about validation, we call this, by the way, like validating the pathway as being relevant for human disease. And drug companies won't go after a target unless it's well validated, you know? So you'll see stuff on the IRG1 knockouts, for example, and they won't make itaconate, and they should be hyperinflamed, you see? And we've evidence, I showed you some evidence for that earlier in a sense. And then the next thing you do is, once you've got good validation, you then got to make a drug out of it. Now, itaconate itself, it's a natural metabolite. You could use that, and that's of interest. More likely, you make derivatives that are more stable and are more drug-like, and we call this PKPD is the jargon phrase for this. And Citrix, which another company I founded in 2016, are doing exactly that. They've made a whole series of molecules based on itaconate that could be more potent, could affect certain tissues, could affect certain diseases, and now we're helping them as well. So that's the kind of journey that you have to go on in drug discovery or translatability. You'd also look at, I mean, for example, at the moment with COVID, we're looking at the IRG1 now gene. So we're trying to, there's variants in that gene in humans, and if we can associate that with disease, that validates that even more, you know. And then you might go after COVID if you can get that validation. So it's all those kinds of things is what we're active in. NLRP3, I mean, that was a good 10 years of validation, you know, in diseases and knockouts and I think our papers made a difference because you could put a drug into a mouse and protect it, you know, that targets NLRP3. That gave strong validation. So it's very similar. The scenario that I've described was happening with NLRP3 in the past.
Brilliant, thank you. Sorry, I'm just checking. We might just have one. Sorry, time for it's been a long day. We might just have time for one more question. So you regularly appear on the news and radio explaining current scientific topics in a way that the public can understand. Is there a need for this wider scale public understanding of medical science and how we go about doing this? You know, maybe.
There is. Yeah, I'm not answering that. This is supposed to be my science day. Hang on a minute, not the media day. No, I haven't to answer that. It's essential. Everybody should do it, reading. I know I do a lot of it, but I think we're obliged to as scientists, to be honest, and physicians, because there's a massive appetite, number one. Number at the moment, especially, of course. Yeah. Number two, you know, you're bringing your expertise to a wider audience. Why not share your knowledge? I lecture to students. I mean, my journey on this began lecturing students, and then I began lecturing the public. It's no different, really. You know, it's a bigger audience. So that's, I think it's really important. The thing is, of course, the taxpayer is often funding your research. Like I said, you want to tell the taxpayer why you're spending the money, you know, so you're obliged for that reason as well. Now, of course, I would say as well, it's not for everybody. And if you can't do it, that's fine. And some people don't like it, or they feel they aren't good at it. That's okay. You know, we all have a role to play. If you feel you're good at it, though, and you want to do it, you should definitely do it. Because I'll tell you one thing, it's massively rewarding. I mean, especially with COVID, I get a lot of people saying, "Oh, thank you very much. You're making something clear for us," you know, explaining something, and it reassures people. There's massive fear out there the whole time, remember, as well. So I think it's really important. The trick is to just be trying. Tony Fauci, who you probably all heard of him, he's the great immunologist. I know him. I've met him over the years. He said 3 things recently about this. He says, first of all, try and be clear. That's an obvious one, right? He said, secondly, if you don't know the answer, say you don't know. Okay, that's really important. And the third one is only use data. No scientist should be in the media saying things unless they can back it up with data, because we're scientists. Try and not stray into politics. Now, we get asked about politics, so we can't avoid it. Try and not stray outside science, because your job as a scientist is to say, I'm telling you this because here's the data. You know, just go on the data. And that's a really good guideline, I think, in general for this media stuff.
Absolutely brilliant. So with that, we might have to leave it there. But Professor Neill, thank you so much for joining us here today. We're really grateful and for taking us through your journey. It'll be really interesting, and we'll be definitely keeping an eye out to see the developments there. And also it'll be really interesting to see the impact that you're having, you know, on, I suppose, Ireland and, you know, young minds or minds of all ages who are looking as well to pursue a career in advancing human health and look to you as an inspiration. So thank you very much.
Great. Thanks for the invitation, Maeve. All the best.
Thank